Welcome everybody. I am Dr. Manar lecturer of medical parasitology. Our topic is cryptosporidiosis and cancer. Our objectives: introduction, association between cryptosporidiosis and colorectal cancer, obstacles, and conclusion. Let's start with an introduction. One of the biggest obstacles to increase life expectancy is cancer, since this disease causes about 13% of human mortality. Normal cells have protection programs, which safeguard them from getting out of control and developing cancer. Oncogenic pathogens has been already recognized by the International Agency for Research on Cancer. These oncogenic pathogens can sabotage this program in order to favor survivor transmission and avoid the immune system. When anti-cancer barriers are altered by a pathogen for its own multiplication and survival strategy, cells will then be transformed into neoplastic cells. In addition, there are several other mechanisms through which pathogens can transform cells into cancer cells, such as the induction of inflammation, chromosome instability, and translocation. Among these oncogenic pathogens is cryptosporidiosis, which is able to hijack the host cell machinery, leading to a transformation of the host cell. As we know that cryptosporidiosis is caused by Cryptosporidium species. These parasites invade the gastrointestinal epithelium of many vertebrates, including humans. Cryptosporidiosis is associated with diarrhea, especially in children, and the causal parasites could be opportunistic and cause life-threatening enteric disease, especially in immunocompromised individuals. Several reports have linked cryptosporidiosis to digestive cancer, especially colorectal cancer. Our evidence for the association between cryptosporidiosis and colorectal cancer are epidemiological, pathophysiological, molecular, and immunological evidence. Let's start with epidemiological evidence. Concerning Cryptosporidium, as we know that cryptosporidiosis is currently endemic in more than 70 countries in the world. And the prevalence of Cryptosporidium is higher in developing compared with the developed one. And a very high prevalence of cryptosporidiosis in malnourished children and those living in area with poor sanitation. Plus other groups at greater risk of this infection, such as transplant recipient and AIDS patients. Concerning colorectal cancer, colorectal cancer is a major public health concern, with cases increasing worldwide. It is the third highest leading cause of cancer deaths globally. There are several risk factors which can predispose to colorectal cancer, such as age, gender, unhealthful diet, sedentary lifestyle, obesity, environmental factors, and other diseases such as bowel inflammation. Also, there is a genetic predisposition and hereditary colorectal cancer. From epidemiological evidence, it appears the risk factors for cryptosporidiosis and colorectal cancer don't overlap, as cryptosporidium infection is more common in children, while colorectal cancer is more common in older population. Also, cryptosporidiosis is poverty related. On the other hand, colorectal cancer is more endemic in the developed countries. So, each disease has different drivers, but Cryptosporidium appears to be one of the promoters of colorectal cancer. After epidemiological evidence, let's talk about pathophysiological evidence. Cryptosporidium host interface. During the interaction between Cryptosporidium and the apical surface of the epithelial cell, result in activation of signaling cascades such as phosphodienyl 3 kinase, proto-oncogene tyrosine protein kinase, and GT-base, which activate a complex of proteins such as 1. RHO family of GT bases. These GT bases are regulators of cytoskeleton dynamics, cell morphology, motility, survival, growth, and differentiation. Also considered as a key player controlling the dynamic of actin-based process. So, RHOGT base signaling can contribute to the hallmarks of cancer. 2. Integrin alpha and beta are receptors that transduce cryptosporidium signaling into the host cell and are primary sensors of the extracellular matrix environment essential for cell migration, growth, and survival. And 3. Rhoptry protein 1, which is secreted from Rhopteris and was found to bind the host protein LMO7, which is a cytoskeletal modulator, suggesting a role in epithelial adhesion and in invasiveness of tumors. Now let's talk about molecular evidence. Cryptosporidium infection causes a hyperproliferation, invasiveness, escape from apoptosis, and immunological responses through the manipulation of host cellular pathways. These abilities make Cryptosporidium a potential oncogenic agent involved in cancer development. And the mechanisms that contribute to the development of digestive cancers, especially colorectal cancer, are multifactorial and involve both parasite and host factors. Concerning the parasite, some virulence factors have been identified, such as proteins involved in oocyst disenchantment, parasite motility, host cell adhesion, sporozoite invasion of epithelial cells, parasitophorus vacuole formation, intracellular multiplication, and host cell damage. And concerning the host, potential signaling pathways may be hijacked by the parasite to induce transformation through alterations in proteins commonly involved in cell cycle differentiation or cell migration, such as beta-catenin, adenomatous polyposis coli, E-cadherin, and P53. And this figure showing a parasite virulence factors during its life cycle, during excitation and release of sporozoites, and during adhesion, invasion, and intracellular multiplication and survival in the host. And about immunological evidence, modulation of the immune response and apoptosis pathways by cryptosporidiosis. As we know that epithelial cells in response to cryptosporidium infection produce cytokines and chemokines to attract immune cells to the infection site and release antimicrobial peptides which can kill the extracellular stages of the parasites sporozoites. However, Cryptosporidium can evade the host immune response by different strategies such as 1. Inside the parasitophorus vacuole, the exposure of Cryptosporidium antigens is limited, so the parasite become invisible to the immune system. 2. The key molecule of the host immune defense against Cryptosporidium is interferon gamma. As the parasite is able to evade the effect of interferon gamma by block signal transducer and activator of transcription 1 state 1 production and endolamine 2,3 dioxygenase expression in order to restore the availability of tryptophan, which is an important nutrient for Cryptosporidium survival. 3. Cryptosporidium infection can downregulate the expression of chemokines which have antimicrobial activity such as chemokine CC motif ligand 20, defensin alpha and beta. 4. Downregulation of P38 mitogen activated protein kinase signaling leads to an impaired intestinal epithelial defense against Cryptosporidium infection. And 5. The most important immune evasion mechanism by Cryptosporidium is to modulate apoptosis and epithelial turnover in order to facilitate its growth and allow sufficient time for intracellular life cycle completion. And this occurs through essential role of nuclear factor kappa beta, which can be activated by 2-like receptors, especially 2-like receptor 4. Other anti-apoptotic factors has also been described such as BCL2, osteoprotogerine, and cervivin. So, the ability of the parasite to evade the host innate immunological response by resist the aporegulating expression of interferon gamma, stimulated genes, and downregulating the expression of other key players favor a persistent infection and alteration of the balance towards cancer. This figure showing the immune response of epithelial cells against parasite, and on the other hand the immune evasion mechanism taken by the parasites to live inside the host. And this another figure showing some variants factors and proteins secreting during invasion or proliferation of Cryptosporidium. Let's talk about obstacles. One of the challenges is difficulty in translating what obtained in mice model to human immune system. The invasive process involved in obtaining tissue samples from the intestine of human is a major limitation in carrying out such studies. Variation in clinical symptoms between mice and humans. The period between primary infection and malignant transformation are frequently very long, and the main mechanism by which infectious agents promote cancer are not involving direct mutagenesis, but due to complex interaction between host and the pathogen. And finally, an infectious agent may trigger the initial events of oncogenesis while being absent in the final tumor. Our conclusion: direct causal link between virulent Cryptosporidium factors and the mechanism of colorectal tumor development is necessary. Recent advances in genomic studies, 3D culture models, gene manipulation, and clinical trials involving the application of sensitive diagnostic tools can be adopted to study the association between the two. And the establishment of a link between Cryptosporidium and colorectal cancer will promote public health measures targeted against cryptosporidiosis, which in turn could reduce the risk of colon cancer. In addition, this could stimulate interest in drug and vaccine discovery research, so reducing the number of children suffering from cryptosporidiosis-related enteric disease and mortality. Finally, we recommend more research efforts in this field as cryptosporidiosis and colorectal cancer have become persistent public health issues globally. Thanks.